I missed my whole introduction, but you don't need to hear it anyway. So, okay, so first of all, you always have to justify, for your graduate students in the audience, you always have to justify why you're doing something. So in the previous year, up till recently, I've had to say, because phage has always been a great system for understanding fundamental things like DNA replication, mutation, and in my case, I always justified in terms of membrane protein structure. But now we can justify it for phage per se, because phage therapy is a reality now. We participated last year in the first successful human intervention, and it's in at least legal one in 100 years. And so there are people in this audience who are likely to have to use phage for treating drug-resistant infections. It's now an official policy of the U.S. government to study phage therapy. That's as a result of the 2014 NIAID resistance program, which is in effect. So now we don't have to justify phage. We can say it's actually important. So it's important to know how phage work, and lysis is, of course, very important to them. So these are when I say phage, I'm really referring to a particular type of phage, and that's the large double-stranded DNA tailed phages, which are called the Caudovirales. These are the most, common genetic entities in the universe, at least on the biosphere. Who knows elsewhere? There's estimated to be 10 to the 31st in the biosphere. Just the unbiased fluorescence assays of any environmental sample from oceans to soil, you find 10 to 100 bacterial virus particles for every bacterial cell. So they're very common. The range of genome size is quite long, between 20 and 700 genes, which means up to the largest bacteriophage so far has had a genome of over 600 kb. And so there's mind-boggling diversity for several reasons. One is that they are highly mutagenic, and they mutated 100 times faster than bacteria. And then also they're highly recombinogenic. So when a phage infects a bacterial cell, it converts it into a hyperrect state. So it likes to deliberately trade DNA back and forth with both the bacterial chromosome with other bacteriophages. So they're highly modular and highly recombinational. So there's an infinite diversity available for bacteriophage, essentially for all organisms. And they are now, of course, being developed as antibacterial agents. And we call them antibiotics with brains because bacteria might resist them, but they got DNA and they can overcome that. So this is just a picture of a random sample. They took images and reoriented them, obviously. But it shows you the type. They all have something in common. They all have a icosahedral-based head that has the DNA in it, and they all have some sort of apparatus sometimes it's short, sometimes it's long, but they have some sort of apparatus that what we call a tail. That's why they all call tail phages. And this is one of the most amazing organelles in all of biology, because it can take a multi-million Dalton-sized double-stranded DNA molecule and transfer it across the bacterial envelope in less than a minute with 100% efficiency. There's nothing like that, and it's what's made these things dominate the biosphere, is this ability to put their DNA information directly into bacteria. So this is what got me involved in Phage Lysis in 1975. This is kind of a recreation, but I was inducing lambda. You can induce lambda lysogens very easily. Lambda is a classic phage of E. coli, and you can make them it's a lysogenic phage. So you can make them start growing. So basically, what you have is a bacterial culture that were the same bacterial culture, but this one was induced 2 minutes before that one, 50 minutes ago. So this is what I'm saying is that over a 2-minute period, this culture goes from that OD to that OD. And so this is the only microbiological phenomenon you can see from 5 feet away. And if you think about what you see here, first of all, this immediately tells you that there's very tight temporal regulation and done by bacteria, keeping time on the order of minutes. I've had children who can't keep time on the order of hours. And so on top of that, it's extremely efficient. And on top of that, the total number of genes involved, as you'll see, is 1. So one gene controls the entire temporal regulation. So this made me, knowing this was a biological event, it stuck in my head, and by the time I had to come up with the second NIH grants, we had a requirement in our department to submit NIH grants until you had 2 every deadline. So I said, what am I going to do? And I thought, this is interesting, so I submitted a grant, and just as a hobby, and now I've been working on it for more than 40 years. And I should say, as already Birgit stole my thunder, Dr. Jenny Garrett, who's now the Dean of Science at Durham College, and not so far from here, was the my first graduate student, and she was the first person to clone a lysis gene, and it was the gene S, as you'll hear about it now. And a lot of the discussion in your paper, by the way, turned out to be right. A little bit was wrong, but most of it was right in terms of predictions. So she started the real research on this. So before we go into the story of how the molecules are involved in this tightly regulated temporal lysis of bacteria, you got to know what the problem is. And the problem is the bacterial envelope is extremely robust. Bacteria are free-living organisms that have to live in an environment that can be quite harsh, and as a result, they have an envelope. And we're going to be focusing on gram-negative bacteria, and that includes, and the outside of all gram-negative bacteria have a special lipid called lipopolysaccharide. It's a special lipid with polysaccharide chains hanging off of it. And then the other inside monolayer of the outer membrane of gram-negative is a regular phospholipid monolayer. And of course, the normal biological membrane, like in all cells, is a phospholipid bilayer. So there are 2 membranes. Think of the outer membrane as kind of as an overcoat. It has large holes in it that are due to proteins called porins that have beta barrel structures, and basically anything below 600 Daltons in size can freely pass in and out of the this is called the periplasm. Think of the periplasm between the outer leaflet and the outer membrane and inner membrane as the stomach of E. coli. So nutrients of certain size get in here, they get broken down to small molecules like sugars and amino acids, and then get imported to be metabolized in the cytoplasm. So in between the outer membrane and the inner membrane is the cell wall, which is shown here as a disaccharide polymer. And gram-negative bacteria, there's basically a single layer of this cross-linked peptidoglycan. It has been thought up until recently that this was the sole source of shape and mechanical structure. It turns out not to be the case. As you will see in a minute, the outer membrane is also determines the mechanical rigidity of the cell wall. And then finally, in the inner membrane, there are intermembrane proteins which always have, as they pass through the membrane, they make alpha helixes. We call these transmembrane domains. They require about 20 uncharged residues, and they can have as many as 14, I think, is the record. But all intermembrane proteins are made up of transmembrane domains. There's one other type of protein that is involved in the structure of the envelope, and these are called lipoproteins. You would think that means they're hydrophobic. So it sounds hydrophobic, lipoprotein, but in fact they're very hydrophilic proteins that have a lipid tail that's covalently attached to them. So they hang either from the inside of the outer membrane or they dangle from the outside of the inner membrane, and that will be important in the talk. Now the periplasmic proteins are just normal soluble proteins that have to be secreted to there, and they float around freely. And we've already said that. So there are 2 physical things you need to know about this. One is that there's about, on normal growth media, LB, there's about 3 atmospheres of osmotic pressure from across the envelope between the cytoplasm and the outside of the cell, and that 3 atmospheres is what causes the cell to blow up. As I said, it was thought that the cell wall was what was restraining that. So the other thing you need to know is that in essentially all eubacteria, the inner membrane is energized. There's about 0.2 of a volt positive outside, and that is generated normally. Well, it can either be generated by oxidative phosphorylation. It can also be generated by glycolysis. And in any case, the point is that the living cell will always energize that membrane, and it uses that energy for lots of purposes, including doing work on the outside of the cell, coupling it to the inner membrane. So one of the indicators of cell viability is whether or not that membrane potential is still there. So the cell wall, if you look down on it, is actually kind of doesn't look quite as regular as you'd think. It consists of these oligosaccharides chains, and it's cross-linked by oligopeptide chains. And this is a computer model from, I forget where, Casey Wang, that's right, he's the one who did this, but he did a computer model of it, and the average size, so you basically what you end up with is chains of disaccharides, and then you end up, as you notice, there's a lumen here that is defined by these polypeptide crosslinks and oligosaccharide chains, and so this lumen is smaller than that lumen, but it's bigger than that lumen, or lacuna, lake is what you want to, sometimes we call it that, and so the important thing to note about this is that if you have, and basically the oligopeptide crosslinks go parallel with the cell axis, and the sugar chains go more or less orthogonal to the cell axis. So that's be a little bit important as we go along. And finally, a protein that of average size of 60 kilodaltons can freely penetrate the peptidoglycan, but larger than that is impeded, and so there is kind of a poor limit to this network. Okay, and that's a typical alpha helix. So if you're looking at the size of a single transmembrane main or alpha helix, that's the relative size. Okay, so this is phage biology in a nutshell. Lambda is the best understood organism in terms of publications per nucleotide on Earth. The second closest is phage T4, and then the next 4 or 5 are all phages 2. So let me summarize lambda biology and the number of papers involved in this. So there are 4 pathways that start once lambda is one of these phages that has to make a decision whether to go lysogenic and integrate in the chromosome or go lytic. But once it's decided to go lytic, at about 8 minutes after infection, it turns on 4 pathways. And those 4 pathways run in parallel without consulting each other, full tilt boogie for the next 50 minutes. And 50 minutes later, the cell blows up, 100 particles are released, and about probably 50 or more phage heads that have, and tails that haven't been connected, but you get the picture. So this is a massive parallel high throughput process, and the three pathways are shown here. One is the pathway by which the phage head is assembled, and DNA is packaged into that head. So the end of this pathway is right here with the DNA-filled head that has an adapter on it. There's a second pathway that starts at the bottom of the tail and then makes the tail, and then of a certain length. And then when it's done, you now have a tail. It has an adapter. You have a head. It has an adapter. And then the shortest pathway is a tail fiber pathway, where it involves just 2 proteins, and a chaperone and a tail fiber protein that makes the tail. These 3 things, tail, head, and fibers, come together to make phage particles. And this, one of the magic things is you can have, using lambda genetics, you can have one cell that does, one induced cell that makes tails, one induced cell that makes filled heads, another one that makes tails. You can take the 3 lysates that have no phage particles together, pour them into a tube, put them at 37 degrees, go home and come back the next day, and you have a large number of phage particles when you're done. So you can, and lambda is the only organism that's ever been put together from purified components and generated a living organism, if you think viruses are living, which I do. So, I haven't said anything about lysis, but the point is that these are all completely independent. If you have knockout mutations that eliminate the head, you still make the normal number of tails, normal number of tail fibers, and so on. So there's no regulation, which is a little disappointing, actually. You'd think that it would learn to do that. Now, the lysis thing is also turned on at 8 minutes after infection, and you'll see the genes involved. And so it is a 4th independent pathway, and the wild type 1 lysis at 50 minutes makes 100 virion particles. And so it also is not informed. So if you knock out any one of these, you get no virus particles, but lysis occurs at the normal time. So that's even more disappointing in some ways. You'd think, wait a minute, we shouldn't lyse because I haven't got any phage particles here, but it doesn't do that. And we only, in all of the history of phage biology, we've not yet found but one case where there is real-time monitoring of the progress of infection in terms of determining when lysis will occur. Okay, so these are the genes that you need to know about. The gene that Jenny cloned, which is gene S right here, and cloning in those days was hard. Let's just put it that way. This was 1980-ish or so, and this was a lethal protein, so it wasn't so easy to clone. Let's just say that for simple. So the holin is the master protein that controls the timing, and all of those wonderful timing events we talked about are enforced by the holin protein. So the holin and endolysin are two very common terms you'll hear about if you hear about phage lysis at all. The endolysin is a fancy word for an enzyme that degrades the cell wall. So it's an endo, comes from inside the cell, lysin degrades the cell wall. The holin, as you might guess, makes a hole, as you'll see. Well, the last 15 minutes we'll be talking about spanins, but we won't talk about them right now. So all those genes that we talked about a minute ago, all the head and tail fiber genes are downstream on this very long transcript. The first few genes are the lysis genes. So it doesn't matter where the genes are. So I'll give you all the structure you're going to, well, almost all the structure you're going to see. That's the high-resolution structure of the lambda holin. It's an integral membrane protein. It has 3 of those transmembrane domains I talked about, 20 amino acid hydrophobic residues, amino terminal out, and C-terminal in. And then there is the endolysin. It does have a crystal structure, but that's all you need to know. It's fully active, so that's why its active site is wide open. It's 150 amino acids or so, and it has the transglycosylase activity. If you add that to peptidoglycan, it gets degraded. All right. So it turns out there's actually another protein, and we won't be talking about this. It's called S107, and that protein is a second gene product of the S gene, and it is an antiholin, and it is involved in the regulation of timing. But we do not have time to talk about it right now. 2 proteins are made from the same gene, and the slightly longer protein is it has a different topology and is a regulator, but we won't be discussing it at all. Because even without it, you get timing just a little bit earlier. All right, so just a brief word about holins. So as Jenny cloned the very first one, she's responsible now for this. There are now 270,000 GenBank records. I just looked at the first 50 or so, and there's maybe 3 or 4 duplications. So there's probably a couple 100,000 holin genes have been discovered now. So you started a real cascade there, Jenny. So we classify them into 4 topologies of the ones we found so far. It's too bad we didn't know what we were doing at the start, because this should be class 3 since it has 3 transmembrane domains. This only has 1, but it's natural. So we did them in historical order. So anyway, I wish I could undo that, but I can't. So they're basically classified by their topology, but even within the topology, there are literally hundreds of families, unrelated sequence. So these things appear to have been reinvented many, many, many times. And there's no evolutionary story where you could get these guys all coming from the same progenitor. So we think the evidence is overwhelming that holins have been reinvented. If you think about that, that means that the way they work can't be that sophisticated. They're not going to be like enzyme with active sites. There's only one of these that's kind of important in terms of. So I told you that holins don't get regulated by any information from the infection cycle. Well, there's only one case that's not the true in the famous phage T4, which is the second best understood organism on Earth. The holin has a periplasmic domain that acts as a signal transduction for exactly that purpose. It wants to know how things are going, and if it doesn't get the right answer, then the holin doesn't work. But we won't have time to talk about that. I'd be happy to answer questions about it. But otherwise, holins are just transmembrane domains, and somehow they do their magic function. And this is, I'll just skip to the end here. There are multiple different types of endolysins. So the peptidoglycan has peptide bonds, it has amide bonds, and it has glycosidic bonds. There are at least 4 or 5 different enzyme types that can degrade that, and it just as this first order of approximation, it doesn't matter. Every phage makes at least one of these, and they're all interchangeable, and they're nice. All of them have been crystallized. They're wonderful structures, stories, and they do different things, but they really don't matter. Think of them as the dumb reporter genes of lysis, right? When they get activated and get to their product, they degrade the cell wall. So here's the summary of the 100, about 80 of the publications that Jenny, and when you can reduce 80 publications to a cartoon with 3 symbols on it, it's a little depressing. But a lot of times it's proving things that takes a long time. The ideas are a dime a dozen, right? It's proving it that counts. So we're looking at the lambda infection cycle, and we're only looking at the holin and the endolysin. So all the other stuff, the phage assembly pathway is going on in the background, and you're not seeing that, but that's phage particles are being assembled in the cytoplasm. And here's 8 minutes when everything gets started. The endolysin accumulates in the cytoplasm. The holin accumulates in the membrane, just as showing it as a simple oval. It continues to accumulate, and then suddenly, at 50 minutes, all of the holins decide to make a hole, And that hole opens up a large hole. These are the largest holes known in biology, even larger than any eukaryotic system. And the endolysins are then free to exit through that hole and degrade the cell wall, and a minute later you get lysis. So that's the summary of what we've learned. And all this time, the important thing to note is that during this period, the holin is completely harmless. So the cell there's no effect of the holin on cell physiology, which is kind of the first law of lysis is first do no harm, right? It's almost like a physician's, because if you want to lysis cell, the worst thing to happen is to make the cell sick before you lysis cell, because then you get fewer virus particles. So it's very, very, and that's one of the key things about holins is that they don't do anything until the time for them to do their hole formation, and then you do it all at once. So it's an all or nothing phenomenon. Okay, so we call this event triggering, holin triggering at 50 minutes. The wild type lambda holin triggers at 50 minutes, and it forms these holes in the inner membrane. All right, so now you get to see your first of several movies. It's not quite sure how to do this. Let's see. Oh, yeah. Is it working? What if I do? Yeah, but it's going too slow. I want to go faster. I think if I put my mouse in, I can. All right, so, whoops. All right, let's just do it now. I can now watch these cells. Whoops. Okay, there's the first one is lysing right there. And this is in real time, so you can see how fast it happens. The thing I wanted you to note is that right about there. Okay, well, if you I don't want to spend too much time on this slide. The cell blows out its contents. You can see it goes to a non-refractile state, which means all of the contents have been expelled. And in fact, the phage particles are ejected as a result of that. And we think that's actually important in terms of the infection cycle. But the cell maintains its rod-like shape for several seconds after lysis. So lysis used to be thought of, well, the cell wall gets degraded, and then you blow up. Well, that's not what happens. What happens is a part of the cell wall gets degraded. The rest of the cell wall is unaffected. And then there's a blowout. We call this local blowout. And actually, I think the next slide is even better. This is a new. That hurts, right? I'm sure that hurts. So the important thing to note about this is that means that there's if you remember our cartoon where the holin forms the hole, the natural Occam's razor model is that this blowout is occurring across from when that hole is because the cell wall gets inundated with endolysin, and then the outer membrane is exploded by the outer pressure. Question? This is real. That's in hundreds of seconds. So this is seconds. 6 seconds, 7 seconds, 8 seconds. We just got this toy last year, and we're having a lot of fun with it. I can watch that movie forever. So in collaboration with Andres Holzenberg, Christos Zava, and his group, a few years ago, 7 years ago now, time flies when you're having fun, we were able to, if you eliminate all the other lysis genes, like the endolysin most importantly, then everything happens normally, the hole forms, but nothing happens after that. There's no lysis because all you've done is form the hole in the membrane. And we were able to use cryo-electron microscopy to actually visualize the holes, which all the reviewers in previous papers said never could possibly exist. But in fact, they do. I just go to the end here. There it is. That's a false color, but so this one's only about 80 nanometers. The average hole size is something over 300 nanometers, which makes them the largest known hole formers in the universe. The largest we've seen so far is 1.5, not nanometers, microns. What am I saying? So no, it's 300 nanometers, and it could be up to 1.5 microns. Think about the size of a hole in a membrane that's 1.5 microns in diameter. So this is a triumph. We sent this paper in. They said, sure, but show us another one. A year later, we finished the second one. So doing cryotomography on E. coli is hard because it's too big. It's a problem of being too big. Oh.
So the whole happens happens not just at holes, and it happens
We'll get that. We'll get to that. Most often they happen at the poles for lambda, but we'll see that in a minute. So this is the top-down view of the lambda triggering model. So what happens is the holin, it actually accumulates as a dimer. So we showed that as among the papers that in that 100-paper series. But they float around in the membrane as a dimer with no effect on the physiology until you get to about 3,000 molecules. And then suddenly they form these, what we call rafts. And we think that that's the secret of this, is that when the rafts form, they exclude lipid. Protein is a poor insulator. And so we think the membrane potential collapses. And as a result, you get hole formation. And we know that membrane potential is key because if you deliberately collapse the membrane potential using dinitrophenol or any energy poison, you go directly to this hole. So the lambda holin will instantly trigger, and it's using it's self-triggering by, we think, the raft causes leakage of protons and collapse. The membrane potential goes 30% lower than normal. That will trigger the lambda holin. Most holins are that sensitive to this. This is one of the most sensitive membrane potential sensors known in physical chemistry. So that's all been published, but I thought I would emphasize that. So this is what we're looking at. Is we think that the hole is 1 molecule thick. So the holins, they go from being a really tightly packed array to a single, to a hole that has a single molecule all around it. So this comes down to actually an idea from Karana's lab for the purple membrane, and that is the critical concentration. So in purple membrane bacteria, the purple membranes start forming when the bacteria rhodopsin reaches a certain concentration, and then once they do that, so all the timing is very simple. Every holin protein allele has a critical concentration, which will go from that to that. So it's actually much simpler than we had originally thought. Now that's regular holins, the one that Jenny, the lambda holin, was the one that Jenny first cloned. It turns out that there's just as many of another type, and they're called pinholes and SAR endolysins. So they're still holins and endolysins, but they're completely different in the way they are. And you can tell from the terminology, the pinholes probably make smaller holes, right? So you'll see what that means. So it turns out that phage 21, a closely related lambda, had a holin gene and endolysin gene. We won't worry about the spanins at this point. But when we actually tested them, it only had 2 transmembrane domains instead of 3. And the endolysin, surprisingly, had a transmembrane domain. So that was very surprising, because this means that it starts out already secreted into the periplasm, but tethered to the membrane by a transmembrane domain. So now, everything else is still operationally the same. So what happens is the pinholin accumulates in the membrane as dimers, and then the first transmembrane domain actually has to come out of the bilayer, and that activates the holin, the pinholin, and then it can start. And when it forms a multimer and triggers to form its rafts, we think the story is still the same. It depolarizes the membrane, and now instead of forming a large hole, it makes a small pinhole, about 1,000 of them. Instead of 1 or 2 large ones, it makes about 1,000 2-nanometer holes. This is Karen Fleming at Hopkins helped us model this using cysteine accessibility studies. And so that we think is a pretty accurate rendition of what the pinhole looks like. So now, why does this work? Well, it's because the SAR endolysin is already out and doesn't have to be secreted. And so the SAR endolysin accumulates in the membrane completely harmlessly, and I've drawn it with the active site shut because the membrane tethered form is completely inactive. But when the membrane potential is collapsed by these 1,000 pinholes, it comes out of the membrane roof holes and becomes active and attacks the peptidoglycan. So we call them SAR endolysins because they have a signal to be exported, and anchor because they're anchored, and they're released. So signal, anchor, release. It was also a way of trying to get SARS research money at the time in case they weren't paying. It worked, actually. So just a quick, with Jim Sakatini's, our crystallographer colleague at A&M, we've done a couple of these SAR endolysins and shown the structure of the active and inactive form, and there's several different types, completely different regulations. This is a typical one. Here's the SAR domain in the membrane, which you have to model because it's an alpha helix. But then when it comes out, it forms the bottom of the active site and reorganizes the active. So we understand how they are active and inactive. Let's put it that way. In the same way with the P1 one, it involves actually disulfide bond reorganization. So these are some of the most dramatic protein refoldings known in biology, and they're intrinsic to the idea of the SAR endolysin activation. So that now we can draw two cartoons that summarize now about 130 papers. And so the overall model, the overall operational systems model, we were talking about, the strategy is the same. You time it by critical concentration of a holin, and then when it reaches that, it causes activation of an enzyme that degrades the cell wall. But the molecular mechanism of how that timing is done is different in the two. But the bottom line is the same. And in fact, these two systems are completely interchangeable, and any phage that has one can use the other. And here's a picture. If we had been alert, we would have known this very early on, because it turns out that they're not the same at the microscopic level. So watch the left-hand box curve and watch it getting shorter and shorter and fatter and then blowing up in the same way. So why is that? Well, in this case, when the pinholin triggers, instead of having a big hole and letting lysozymes coming out and drenching a part of the cell wall in endolysin, the pinholes are formed uniformly around the cell. It causes depolarization of the membrane, and the SAR endolysins are activated isotropically around the cell. So the cell wall is degraded isotropically, and that's why it tends to round up before it lyses. So, of course, we never looked until after we saw that a molecular basis was different, and so it was very satisfying. The other thing to notice is that only 3 of the 8 or 9 cells that are shown here lysed, and it turns out that the SAR endolysin, pinholin SAR endolysin system is not quite as tightly regulated as the canonical holin endolysins. So there's something slightly less. So the time frame being, instead of being 2 minutes, all the cells lyse, is more like 6 or 7 minutes. So it's not as sharp. Whether there's biological importance to that, we don't know. So this is a summary. We now know there's a lot of stuff going on. So these are we'll talk about spanins here in a minute, but holins and endolysins and pinholins are the functional parts of this system. But it turns out that there are multiple different physiological and molecular regulators of this. And if you think about it, the most important thing that a lytic phage is regulating is its lysis timing. If you think, a very simple allegory, the longer you go, the more phage you'll make. But on the other hand, the longer you go, the infected cells out there might be accessed by another phage. So there's 2 evolutionary pressures that are facing you on that. So it makes it the most tightly regulated all-or-nothing system that's known, as far as I know, in biology. So these are just a graphical summary of all the different regulatory. There are proteins. There are anti-holins in both the membranes and in the cytoplasm. There's there's anti-holins for every holin. The cell wall regulates the spanins, as you'll see in a minute. There's also positive activators, releasins that actually are required to help the SAR endolysin out in some cases, coholins that are required for functional holins, and their environmental information comes in terms of membrane potential and other things. So it's very tightly regulated and is much more complex than we had originally thought. And there's a signal, at least, that we told you about already that we call no quorum signal for T4. All right, so the last part of the talk now, I want to talk about the unexpected players. We thought we had the whole story. There's the pun, okay? The whole story. Gosh, this is a tough crowd. So I usually get a titter out of that, but 2 people are all asleep now. So it turns out there was 2 genes downstream for SNR that their only importance historically was that I discovered them when I was a postdoc at Harvard. And lambda was so important in those days that anybody who discovered a gene in lambda, you got a job. So these things had the very important biological impact of getting me a job. And my wife was ready for me to have a job. So it saved my life and my wife's and my marriage and everything else. So, but since I had everything understood in terms of the whole endolysin, I never worked on it forever. It was called a accessory lysis protein, right? So that was dumb, actually, if you think about it. So it turns out that it's actually 2 genes, and I keep saying unique in biology, and unfortunately it sounds like a repetitive thing, but in fact this is the only case in biology where there are 2 genes in the same DNA that make completely different proteins that are required for the same biological function. So the evolutionary impacts of that are not obvious how you would ever get that way, right? If one gene requires the other, how did that evolve? I mean, furthermore, having 2 genes in the same DNA in different reading frames is very unusual. Most of the known examples are in phage, but in all the other cases, the 2 genes do different things. These 2 proteins are required to make a complex, and they're required for lysis, as you'll see in a second here. So And this is a blow-up of them. So one of them is an intermembrane protein. That's the RZ gene. And it has a transmembrane domain. It's amino terminus, and it's very hydrophilic. The other is a membrane lipoprotein. Remember, one of these very small proteins, it gets processed to a cysteine, and then lipids get attached to that cysteine, so you end up with a highly hydrophilic protein that is dangling, in this case, from the outer membrane. So this is an intermembrane protein, that's an outer membrane protein, and it turns out they make a complex. And so the reason why we want to look at these is we finally got around to making nonsense mutations in these two genes and discovered that lysis didn't occur. 30 years after we've been studying on this, we discovered that these genes are actually required. This was hard to explain in my NIH renewal. Oh, by the way, so don't do that. You know, don't do that kind of thing. So let's see if I can get this started. So if you knock out either or both of these genes, this is what happens. They try to lyse. Instead of lysing, they form spheres. This is very disturbing, right? So why didn't we notice this back in the old days? Well, in the old days, we studied lysis in the shaker flask. We didn't have microscopes. And these are very fragile spheres. So when you're putting in a shaker flask, they just get broken. So we never noticed because but shaker flasks are full of shearing forces, which are not common to E. coli except in people my age in the colon. So you don't want, so most young, I experienced a lot of shearing forces in there, but I don't think lysis evolved for doing that particular, this is a tough crowd. So, so at any rate, looking at this already, if we had done this without knowing about it, what does this tell you? This tells you that the logical Occam's razor model is that the cell wall's gone because it's no longer a rod, but it's not lysed. That means the outer membrane is retaining this 3 osmotic pressure system, and so because we didn't ever look in the microscope, which is we were microbiologists, we should do that. We didn't really know that this was the case. So that's a lot of, and this is also true for pinholes and SAR endolysins. In this case, they would start to round up anyway, but because of the mechanism, but they never lyse. So this is here's an actual cryo-EM again with the Holtenberg group. This is a cryo-EM showing one of these round cells. You can actually, this is one of my favorites because it actually shows the hole with the guts of the bacterium coming out of the lambda. This is S plus, R plus, no spanin. And so you have the outer membrane intact. The inner membrane is intact except for the hole, and there's no cell wall. Normally we could actually see it. So this proves that the outer membrane is capable of withstanding the internal osmotic pressure of E. coli, which we did not know before we discovered the Spanin phenotype. So, all right. So now we have to modify our cartoon. The pathway was incomplete, so something has to happen to the outer membrane that's Spanin-mediated. So the question is, what are the spanins doing? All right. So how do you disrupt? If you know now that you have to disrupt the outer membrane, how do you do that? And remember, you wanted to make it all or nothing. So it's not obvious. If you secrete an outer membranase with the endolysin degrades the cell wall very quickly. It doesn't have to degrade very much of it, just enough to cause a blowout. But the outer membranase probably has to degrade. And how is the outer membranase going to degrade it? What it encounters is a phospholipid layer. You can't tell the difference between the inner membrane and the outer membrane. So we knew it couldn't be enzymatic. We're fairly sure it couldn't be enzymatic. So it turns out, I'll just skip to the bottom line here, that here, this is a slightly more detailed structure. It all comes about because of a mechanical event caused, there's mechanical disruption of the outer membrane caused by a machine built of these two proteins as subunits. So the key issues here are that the periplasmic domain, this doesn't matter, you just have to have a transmembrane anchor. There are two coil regions and a flexible hinge region. And then the outer membrane lipoprotein. Notice that this flexible region more or less corresponds to where this other gene is, and so that's evolutionarily satisfying because you can put any sequence in here that's flexible, and then that allows you a lot more ability to generate another completely different gene in the outer reading frame space. But in any case, the important things to note besides this hinge in the two coil regions is also let's see. that this is highly proline rich. And that was one of our first clues, because those proline-rich oligopeptides are membrane destabilizing. So we think this is actually the payload for disrupting the outer membrane. So I'm at 1:03. When do we get out of here? At 4 o'clock, is that right? I'm known as the Fidel Castro of Phage Lysis, so except he died. So, do what? So what time do we have? Is it 10 minutes? 10 more minutes? 10-ish? That's dangerous to say somebody my age, that's for sure. So here's the simplest diagram. The spanin protein is in the outer membrane, dangling down, and it makes it. We know this. It makes a C-terminal complex with the inner membrane. We call this the O-spanin for outer membrane spanin and I-spanin for inner membrane spanin. And they make a complex by C-terminal interaction. And importantly, this complex, once it's formed, it's very stable, is trapped in the peptidoglycan. Because the peptidoglycan, remember, has these lacuna that are bounded by the oligopeptides and the sugars. And for the most part, these are covalent and stable. And so once this complex is formed, this thing can't go anywhere. It's got to float back and forth inside the peptidoglycan. So it originates in its inactive form because it's trapped in the peptidoglycan. So you can already see where I'm going. It won't do anything until the peptidoglycan is degraded. And then it can do whatever it wants to do. So just a quick bioinformatics. Once we discovered this, the scales fell from our eyes, we started looking to find if other phages had these things, and sure enough, they all do. Since I'm short of time, I'll just go to the bottom line here. There are spanins that have this same one inner membrane spanin, one outer membrane spanin that has a lipoprotein signal sequence, and they're essentially all phages, or let's put 90% of the phages of gram-negative bacteria have these guys. They are extremely diverse. So lambda and T7 have very similar ones. But they're not. There's no sequence similarity, but they have the same embedded structure. Currently, there's 181 of these unrelated families, and they're called embedded because the entire Z1 gene is inside the other one. The entire O-spanin gene is inside the I-spanin gene. There's also a bunch of ones that have the O-spanin that actually protrudes beyond it, and then in fact, in famous phage T4, the genes are completely separated. And so this was very important to find because this is impossible to evolve de novo. I'm sure they all started like this, and there's a very important compression phenomenon in phage genetics, evolution, because phages have a very small space to put all their genes have to fit in what will fit in the phage head, and so anytime you can make things shorter, you get an advantage. So we think that they started like this and they evolved to get that way. And then just as one final fillip, it turns out in the phages that don't have two-component spanins, they actually have and I don't know how many of you here know about phage T1. This is a bad dude. So phage T1 is an airborne phage, and if you don't know about it, I can show it to you. But it means once you have it in your air system in this building, you will not grow E. coli anymore. So this is, I know, Jenny, you went through at least one T1 infection. Once this gets in your building, you're basically permanently, so it's a lytic phage, and it had no spanins, except it turns out it has a unimolecular spanin, which is, and we've now shown that so this unimolecular spanin has a lipoprotein signal as we expected, and has a C-terminal transmembrane domain. So if you think about it, it does this single spanin does the same thing. I won't bother with this when we have enough time. It makes operationally. This is another example of a different strategy to get to the same operational thing. You end up again with a protein bridge between the outer membrane and inner membrane. Only now it's a single covalent polypeptide and not a C-terminal complex. So that's the common thing, and we think that they do this. As you'll see in a minute, we think we know what they both do the same thing, and that is they're going to fuse the outer membrane with the inner membrane. And so in order to fuse the two, you have to be able to grab both membranes, and in fact they do it. These are not evolutionary related, right? These are completely different events, but they do the same thing. Okay, so it turns out that our structure, Texas 3-step, our structure was a little simplistic. These guys are actually dimers, and they're linked by disulfide. So instead of being one O-spanin and one I-spanin, there's a covalent dimer of the O-spanin and a covalent dimer of the I-spanin. So it's just a little bit more complicated molecule, but otherwise it's the same thing. Okay, here's the model that you can take away from since we're almost out of time, the Texas 3-step model. So we think this part is very well established now, that the triggering causes the formation of, we're looking at the canonical holins. That lets the endolysin out. The endolysin then degrades the cell wall, and then that liberates the spanin complexes to do lateral diffusion, and that we know that their periplasmic domain oligomerizes into a 25 nanometer rod in vitro. So we've done that. We've published that. So we think this is what happens: is that they oligomerize and then collapse to fuse the membrane. So we did one experiment to test this, and then it worked. And so we're never going to test it again. So the important thing is, this is not an enzymatic solution. This is a topological solution to this situation. You're instead of destroying the outer membrane, you're making it irrelevant, because you're creating a. If you notice what's happened here, you're getting a continuum between the cytoplasm and the outside wall. And if you suddenly go with a 3 atmosphere pressure here, you suddenly create this. You can see why that would be a catastrophe. You're going to instantaneously have to release through. The only thing, fluid flow is the only left limit on how fast this is going to occur. All right, so here's the two cartoons drawn with a little bit more detail. That's for the 2-component spanin, and this is for the unimolecular spanin. The reason why we have different cartoons here is that there's no there are only beta sheet domains in the unimolecular spanin. So we don't. This is all hypothetical, but we think they're making a beta barrel type of complex, whereas these guys are definitely coils, and this is a very common membrane fusion topology in eukaryotic viruses. So how to test it? So this is the last experimental slide. How do you test it? All we really wanted to find out is whether or not these proteins were capable of fusing the inner and outer membrane. And it's very difficult to make. In fact, no one has ever been able to make an artificial outer membrane because of the two-layer structure. So what we did was took advantage of what's known as spheroplast technology in E. coli. And so what you basically do is that you can use EDTA to disrupt the outer membrane, and then hen egg white lysozyme to degrade the peptidoglycan. And you basically create a spherical cell. Now you have to do this in sucrose, otherwise the cell will blow up from the 3 atmospheres of osmotic pressure. So basically the outer membrane, the cell wall is degraded, the outer membrane rolls up into little nurdles, and you have basically the cell, I don't know, should be spherical. Does it look like an egg? It shouldn't be an egg, it should be a spherical. I don't know how that worked. So if we do this with a cell that makes the intermembrane spanin, everything is going to be just what we want. So the intermembrane spanin will be made. It'll be projecting from the surface of the inner membrane. It'll be a dimer because that's the way it accumulates, and it no longer will be inhibited to the cell wall because there is no cell wall. We had to do a little bit of a trick. Whoops. Why is it going backwards? Oh, all right. So we had to do a little bit of trick for the O-spanin. So in gram-negative bacteria, one method by which lipoproteins are exported to the outer membrane in one pathway by the so-called LOL pathway, and you don't know the details of this, but so basically a protein, there's an ABCD transporter that takes the new lipoprotein, transfers it to a shuttle protein, which then transports it to the outer membrane, then it dives into the outer membrane lipid. And this is done for all of the outer membrane lipoproteins, but this machine has a way of making sure that it's not inappropriately sending intermembrane ones. It checks the first 2 residues after the cysteine, and if they are anything but asp-asp, then it goes to the outer membrane. So all we had to do to redirect RZ1 to the intermembrane was to convert the threonine-serine sequence to double aspartate. And that meant that it would no longer enter this pathway and would accumulate in the outside surface of the inner membrane rather than the inside surface of the outer membrane, which is exactly what we wanted. So in effect, we call this IMRZ1, and it's normal in every way except it has 2 aspartates right after the cysteine in an area we had already shown was irrelevant sequence. It could be a linker sequence. So the experiment is pretty simple. Then you make 2 types of cells. Some cells expressing RZ, and you make spheroplasts out of them, or you make cells expressing this modified RZ1, and you populate them with either GFP or mCherry, and then it turns out that if you have that situation, you get fused spheroplasts with both color. And of course, all the controls were done in the sense that if you knock out either one, you don't get the fusion, and so on. So. want to get to a movie here if I can. So this is just some of the raw data from this paper that was published 2 years ago now. And so the fused spheroplasts turn out to be irregular in shape, and I don't know whether that means anything, but that's probably we get about 10%. And so this is just let me. I thought I had a. Oh yeah, this is the movie. I wanted to show you the movie. Is that not going for some reason? Oh, okay. So unfortunately, this one didn't. Yeah, they tend to. Sometimes they lyse pretty soon after. Right? There's something bad about being suddenly twice as big as you. Let's see, and then of course it doesn't happen with spanins. They just stay that way. And then more importantly, I'll just skip now to. We had a lot of genetics. If you can't do genetics with lambda, you really can't do genetics. So we had lots and lots of mutants, and they turned out to be useful. These were all missense changes. They had no effect on accumulation or localization, but didn't function in fusion. And luckily for us, when we incorporated these into our in vitro assay, wonderfully the proteins stopped exactly where they stopped exactly where we expected. So you ended up with membrane spheres that were forming the complex, but fusion didn't occur, which is really nice. So it says that these defects really are in that last step of the membrane fusion. Okay, so and this is another movie in real time. That one blew up. Wait. So at any rate. Okay, so there's the summary again. And then I'm going to show you a couple more movies since we're done. Think of this as the cartoons after the show, right? So you get to. So this is all preliminary unpublished stuff. This is, again, lysis at 100 frames per second. So it's, in that case, the whole form right there, and you can see all. Remember, phage particles are coming out of here, and you just can't see them. And then gradually, because the endolysin is spreading down the periplasm, it degrades the rest of the cell wall. But that's like embalming the corpse, in effect. This is real. This is seconds here, so it's much slowed down. Now, we wanted to know. So, if you believe this model, I actually didn't believe it. I'll be honest with you. The people in my lab had to convince me of it, but now it's my model, so it doesn't matter. So, I think I did the same thing with Jenny, but at any rate, models are there to be tested, right? So, my models have always been shown to be wrong. So, I'm advancing science that way. And this model, I think, is correct. But so, one prediction of this model for lysis is that the periplasmic contents never participate because they get walled off. All right. So what happens is what we did is we put superfolder GFP into the periplasm with the signal sequence. So it's now you're only seeing that, and then we knocked out the spanins and we did the lambda induction, and so the prediction is that they'll stay segregated all the way through lysis. And of course, they don't lyse because they're spanin minus. And now these GFP is gradually leaking back into the cell through the holes that are formed by the holin. I just love this picture. This is just like magic to me. Now I think the $100,000 we spent on this was actually worth it. So, and here's where we actually allowed the whole lysis, the same experiment, only now you let the spanins do their thing, and it's, I think, even more convincing. Let's see. Whoops. I've learned all sorts of stuff about backgrounds and that I didn't know. Okay, so it's about to blow, I think. Let's see. So you end up with all of the periplasmic contents sequestered into little spheres, and those spheres are half inner membrane and half outer membrane. Which I think it's cool. All right, and is there anything else? Okay, this is following. I asked him to give me some pictures to show as cartoons, and here's one where we actually tagged the holin with the GFP protein. This is very preliminary, but in fact, I just saw these last week. So the holin is only made in like 1,000 copies, so there is a problem with background fluorescence, but I think you can see that the holes form, the rafts form at the poles suddenly. So they're all free floating until all of a sudden, now you can see this time delay up there, all of a sudden they're collecting at the holes. And remember that the concentration of protein did not change at all in that 5-second period. So it's not an overproduction phenomenon. This is a redistribution that happens on the scale of seconds. And then Now you notice they disappear and then you explode. So why do they disappear? Well, think about what's happening is first you form a raft where it's an area that's all GFP, and then they expand out to form a ring that's one molecule thick. And at our resolution, we can't see that. So that's our story, and we're sticking to it. Did this one here? I think this is another one. This is where sometimes it doesn't happen at the poles. But you can see rafts form, and then a little bit later, after the rafts are formed, then you'll see blowouts at those positions, I hope. Do something. They disappear because they're forming the holes, and then you should see little blowouts at all of those three positions. Yep. Okay, I think I'm done. So I want to say that the Spanin story, current students are Rohit Kanguri. Jesse Cale just graduated, so he's now a postdoc in the laboratory. I'm down to 2 students. When I'm done with them, there'll be 33 PhDs, and she was number 1. And her work actually supported about 30 of them. So Jenny, you really get a lot of the credit for this. And we also like to thank Johnny Football for wearing the lambda on one of his last games. And, thanks for your attention.
Thank you, Roy, for a wonderful story. We have time for a couple of questions.
Yeah.
So membrane voltage collapsing and then endolysin getting activated. Can you do that like chromophore?
Yes.
You can.
Now remember, in the canonical system, the enzyme is actually already active. It's inside the cell, fully folded, fully active. So the activation just means getting to the periplasm. In the case of the SAR endolysins, the ones that start off outside the cell but are inactive and tethered to the membrane, you get exactly the same result if you add dinitrophenol or cyanide. They come out and they activate. So in that case, they don't have to go out because they're already out. They're just inactive. So these SAR domains are stable in the energized membrane, but they come out as soon as in membrane. And oddly, they require essentially complete depolarization, whereas the holin triggering only requires about 30%. So you can Howard Berg has this really nice system where you can titrate down. You tether a cell and you can measure the spinning rate, and that's a direct readout of the proton motive force. So you can titrate down that with dinitrophenol and show that at 30% reduction, that's when the holins will trigger. So, and we actually did an experiment with Mike Manson where we put the holins in a cell that was spinning, these tethered cells, the Howard Berg experiment, and they spin normally. You turn on the holin genes, it's normal, 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 stop. So we actually titled that paper "Holins Kill Without Warning." So that was his title. Yeah, generated over many beers. So that's yeah.
There's a lot of membrane fusion events that occur in biology.
Yeah.
One of the envelope viruses in a million cells. Is there any Spanin-like type things that?
Yeah. So our cartoon with the coil domain, that's very reminiscent of the what's it called? I'm blocking on it. It's the fusion protein for HIV. Somebody here must know what's it called. It's the one, but that's used to go in the cell rather than coming out of the cell. So it's essentially the same notion. There's a collapse, coil-coil interactions, and that brings the two membranes together. Then something still has to destabilize one of the leaflets, and then you get this hemifusion event and so on. And when I gave this talk at Biology International at Basel a year and a half ago, they all came rushing down and said, and the nice thing is we have genetics for this. You can't really do genetics very easily on membrane fusions and eukaryotic viruses. So it's the same story, I'm pretty sure. Yep.
For the transition from initially you have the whole story, you have accumulation or a distribution of the holins on membrane, and then they form these rafts. What are regulating the transition from just a concentration effect? Is there something else in there?
We think that in the Corona theory, he did it with bacterial rhodopsin. Okay, so we have genetics that I have, and Jenny generates some of these mutants, but we have genetics that mutate single missense changes in the transmembrane domains of the holin, right? So just alanine to valine, right? Nothing spectacular. And every one of those changes dramatically changes the triggering time. So you can go, the earliest we've ever seen is 9 minutes, which is 1 minute after, which is very bad because you've lysed the cell before there's any phage particles, out to almost 2 hours. So one amino acid change will make you will trigger at a different time, and that means a different critical concentration. He already showed that with bacteriorhodopsin. Bacteriorhodopsin has 7 transmembrane domains, and it was already known from the low resolution structures of the purple membrane where the interaction places were. And so using that information, they went in and did site-directed immunogenesis in bacteriorhodopsin to change the residues at the interaction sites, and was able to do exactly the same thing, change the critical concentration by tenfold up and down. So we think it's an exact model. Now, I also think that there is a component of membrane curvature, because it looks like that the holins that tend to be polar holins, like the lambda does most of the time, there are some that are and some that aren't. And we think that what happens is the curvature at the poles activates the holin for triggering. We have several different reasons for that, but mostly it's the critical concentration. And the beauty of that is evolutionarily is if you're a phage, you go through in a week, which is is an eon from a phage point of view. If you, especially let's say if you're in a phage out in the dirt somewhere, so you're going to have periods where there's lots of hosts and periods where there are no hosts. And so according to evolutionary theory, that was actually worked out by a former postdoc of mine, Ying-Nong Wang, before he was a postdoc in my laboratory. It turns out that the ideal phage wants to match its lysis time to the fecundity of the host. All right, so that means that if there's a lot of particles out, a lot of potential hosts out there, you want to have a shorter lysis time. You'll accept fewer phage particles by quicker access to all these bacteria because they may not be there very long. On the other hand, if there's almost no bacteria out there, you want to stay in the cell as long as possible, get as many phage particles as possible. In order to do that, you can either do real-time regulation of the holin, which we know, unfortunately, only one case, and that's T4, which is the way they all should do it, but T4 does it that way. But in all other cases, it looks like the phage just rapidly evolves to the right time. And you can do that rapid evolution because a single mutation, essentially anywhere in any of the transmembrane domains, if it doesn't inactivate the holin, will change the lysis time dramatically. So you just, phages have 300 progeny, let's say, or 200. It doesn't matter if 10 of them are funny-looking kids. If one of them has a later lysis period, and it might turn out to be the one that gets expanded. So there are pre-existing mutants because of the nature of the holins. In any phage population, there are already pre-existing mutants that are absolutely perfectly attuned to whatever new fecundity system you have, either high or low bacterial cells. And so holins are interesting because they appear to have been selected for their evolvability, for their rapid evolvability. And they've been invented multiple times. We think, and because I don't think they do. In fact, I think it's harder for membrane proteins not to be a holin. So we think membrane proteins first. If you become a membrane protein, if you don't watch it, you're going to end up being a holin-like protein in aggregate, and then that's bad for the cell. That will cause membrane depolarization. So essentially, all normal membrane proteins have to make sure they don't oligomerize into big rafts, because proteins would rather, in the final analysis, every transmembrane domain would rather be with other transmembrane domains and not be with lipids. So you have to evolve the ability not to aggregate, and I think that's been shown to us unfortunately, when you overexpress membrane proteins in the E. coli, they almost always aggregate and become useless, right? So they've been prevented from doing this. So I think the answer is that it's mostly critical concentration. That's a long answer to a short question. Yeah.
So brought up the point of the evolution. So some bacteria have they evolve quicker or slower than other bacteria. So the phages that affect those, their replication mechanisms affect the rapid evolution of the phage?
All I know is that in all the cases that I know about, the phage replication has about, I don't think the answer to your question is known, but the mutation rate per replicated base is 10 to 100 times higher in phage than it is in bacteria. Partly, in some cases, like lambda uses the replicon of E. coli. It just repurposes it. It doesn't do anything but say, this is not the E. coli origin, use my origin. So you'd think that it would have the same mutation frequency, but it squelches normal repair systems. So it deliberately squelches it so it mutates at a higher frequency. RNA phages, I haven't told you about, they're they mutate at 10 to the minus 3 per nucleotide per generation. So basically, RNA phages are just like mutagenesis machines, and they also have, you know, they've chosen a completely different way of lysing the cells. But in every case, the evolvability of lysis genes appears to be an important criterion. I don't know the answer to your question, though, whether or not in high, which bacteria have low mutation rates? Or they, you don't know of any?
I know the alphas are sometimes
Are they, they're slower mutation rate? Did not know that. Yeah.
I'm a philosophy biology, and this is maybe too speculative, I'm not sure.
Nothing's too speculative.
But the process from T4 with 2 or 3 genes that were separated.
2 genes.
Well,
The two spanins.
The question what serves as the hinge to.
Yeah.
Two
They have the same. They have basically the same predicted.
shift.
No, there's no frame shift. So I should have emphasized that. So these genes, there's no frame shift in this. These are 2 genes in the same DNA, so they make the same one messenger RNA, and the ribosome either translates one or translates the other. They're in different reading frames, but there's no frame shift.
I see.
They're completely.
But there is a frame shift.
No.
When you in the case in which you have an embed
There's no frame shift.
no frame shift.
There's lots of frame shifts in phages, but this isn't one of them. So just think about this. Here's a messenger RNA. You're a ribosome. You're a 30S ribosome all ready to initiate. And it turns out there's 2 Shine-Dogarno sequences. One's here, and that gives you the full-length guy. One's here, it gives you the shorter guy in the other reading frame. So there's no frame shift. The ribosomes.
Even so, the question remains: is the invention of the intron perhaps modeled, but without the frame shift?
But it's not an intron either, right?
Well, what's serving as the hinge?
Well, the hinge. So the hinge. We know it's a hinge because you can replace the 30 residues in there by glycero. You know, so we know it doesn't matter. But and that's true for the ones that are separated. They also have the same structure.
That's a mechanism for excising.
No, they don't excise. The protein is made as programmed in the messenger RNA. You either make the 150 amino acid intermembrane protein or you make the 60 amino acid outer membrane protein. There's no intron. These are 2 completely different DNA sequences, messenger RNA sequences in the same DNA. They're just different reading frames is what they are. But to answer your question, even the ones that separated, the T4 one where we think they all started, they also have the hinge. It's just it's encoded. There's no other DNA, no other genes under the hinge. It's just by itself. Yep.
I have one question. So, how do the phages decide which sites in the membrane will be broken?
How do they decide which sites to?
In the membrane will be broken.
I think the lambda holin tends to trigger at the poles about 80% of the time, but otherwise it's random. It doesn't care.
But from the movie you showed to us, it seems they always break the terminals.
The poles. Yeah, I showed you. Well, I showed you 3 movies, and 2 of the 3 showed polar lysis, and 1 of the 3 did not. So, anytime you're seeing movies, remember somebody chose that movie, right? So, if it's a crappy movie, is it because I'm being honest and showing you that on average we have crappy movies, or is that the very best I can do? So I think that anyway, the lambda holin trigger seems to do it at the poles about 80% of the time. Other holins that we have looked at don't. Oh, why they do it at the poles more often? We think that there's a curvature effect that potentiates the triggering, but we're just guessing, and it's not going to be easy to test that model. So, yep.
On the pinholes,
Yeah.
how many pinholes?
About 1,000. There's 7,000 molecules. We very carefully measured it by quantitative Western blotting, with very careful purified protein as a standard. And so it comes out to the wild type triggers at 7,000 molecules and makes 1,000. We know that they're all in pinholes because you can do the cysteine accessibility and show that every single molecule is showing that cysteine to the lumen. So they're all in pinholes. There's possible that they're not all 7. Some might be 8, some might be 6. But when we do cross-linking, that 6 is what we see most, or 7 is what we see most of the time.
And how many pinholes are necessary for that?
That's a really good question. So one movie I didn't show you was, I told you about so Howard Berg, the great physiologist, has an experimental system where you take E. coli and you basically tether it to the glass slide by its flagellum. And normally the flagellum turns in order for you to swim, but if you're tethered by an antibody to a glass slide, instead of you swimming, you go round and round, right? So this is the beauty of this system is that it's a non-invasive direct readout of the proton motive force, and you can show that from the maximum down to the minimum, you can titrate it out with the protonophore. The velocity of the spinning, we call it the spinning bug model, the spinning bug thing, the velocity is exactly proportional to the proton motive force. It's still my absolute favorite experiment to do. And so when you do it with the canonical holin, I think I already told you what happens. You turn on the canonical holin, and 30 or 40 minutes later, depending on how level of express it, it's normal spinning, which means no toxicity, and then suddenly they stop because it's triggered. But with the pin holin, it spins and then stops, and then starts spinning again. And then each time it stops for a longer period of time, over a period of about 4 or 5 minutes, and then it's dead. So the cell is fighting back. But we don't know how it's doing that. So we don't know how it's doing that. Now, I would show it, but the first thing you would ask is, why is that happening? And I have no idea. So it obviously can't fight back for a hole that's a half a micron wide. That's your toast at that point. But when you're talking about, it's possible that not all the pinholes form at the same time. Maybe you only make 100, and the cell has some way of either spewing out more protons or plugging these holes up. That's the only thing I don't know. So that's the difference. All right. Yeah. Sorry it took so long. Okay. No, that's good. All right.